All right, welcome everyone to this session. Good morning, or good evening, or good afternoon, depending on where you're joining us from. My name's Matt Ritchie, and it's a pleasure to welcome you to this session at BIOC 2021 on education and frameworks. We'll, while we wait for people to settle in and get connected, we'll start with a few housekeeping items. The first is that if you have questions for the speakers, please add them to the Q&A tab. People have the ability to upvote questions that they'd like to hear the speakers answer, and the questions should appear on the stage. If you'd like to ask a question live, you're very welcome to. Please use the raise hand feature and the moderator will bring you on stage. And also don't be shy, there's lots of emoticons at the bottom of your screen. Feel free to rain the speakers with praise, with different emoticons as you feel fit. Another great feature of this session is that the talks are all recorded and they'll be available online shortly after the session. So if you have to leave or you have friends who you know are attending other sessions, they can all, of course, catch the content of this one at a later stage. So it's a great pleasure to introduce the first speaker, Charlotte Sonnerson, who recently took home a Bioconductor award. So congrats for that, Charlotte. Charlotte's going to be talking to us about the Bioconductor Teaching Committee and that new initiative. So welcome, Charlotte.
Thank you for tuning into this talk, where I would like to give an update about the activities of the Bioconductor Teaching Committee. My name is Charlotte, and I'm speaking here today on behalf of the whole committee. So the Teaching Committee was established last year, and it's currently organized under the Bioconductor Community Advisory Board. It's co-chaired by myself and Laurent Gatteau, and it's really open to any member of the community who is interested in bioconductor and education and training. And in the end of my talk, I will give a little bit more information about how you can join and engage with our activities. When we established the committee, it was with several different goals in mind. First, we wanted to provide a community for people interested in training and bioconductor, and also a place to share experiences and coordinate training activities. For example, last year, 5 community members had the opportunity to become certified carpentry instructors, and we plan to offer more such opportunities in the future. We were also very interested in establishing a broader connection with the Carpentries. So the Carpentries is an organization that aims to teach foundational, computational, and data science skills to researchers, and also to collaboratively develop open training material, all based on sound pedagogical methods. And the Carpentries already provide several introductory R lessons, but none of them is specifically focused on what Bioconductor can offer. So our second aim was to start assembling bioconductor-focused training material, which was still consistent with the carpentry structure and guiding principles. And finally, we would of course like to coordinate the delivery of this development training material in various bioconductor-related events, such as conferences like this one or standalone courses, both online and in person. As you can see on this slide, there are already many community members who have engaged with the activities of the committee, which is really great. And I would like to take this opportunity to thank everyone who has contributed to taking us where we are right now. And in particular, I'd like to highlight the great support and practical guidance that we have received from the Carpentries, in particular from Toby Hodges. So at the moment, we have started assembling an initial set of 3 lessons or training modules. And these lessons are being developed openly in the Carpentries incubator on GitHub. So you can follow the links that I put here in the slide. The material will be available for public use under a CC BY license. And we also encourage people to, of course, contribute if they would like to. So the BioC intro lesson provides an introduction to R and data analysis with a focus on omics data. It also introduces a small set of bioconductor objects, in particular the summarized experiment class, but the focus is really on basic R programming. The BioC project lesson instead is fully focused on bioconductor, and provides an overview of the bioconductor ecosystem, both in terms of the standard classes, but also shows how to get help and navigate and interact with all the available resources. And finally, the BioC-RNA-seq lesson is intended to provide a conceptual introduction to RNA-seq analysis with Bioconductor. So I mentioned that these lessons are developed according to the Carpentry's guiding principles. So what does that mean, and how do we adopt these principles in the lesson design? Well, what we would like the learners to come away with is a set of foundational skills and concepts, and a framework or a mental model that connects them, that they can use to continue learning and build upon beyond the workshop. So to achieve this, we employ a backward design when building our lessons. We start by identifying the target group and the desired learning outcomes, and then use these to design exercises and to plan then the rest of the teaching material. We also attempt to keep the cognitive load low by introducing a few concepts at a time, and also including many exercises that are also carefully designed to help learners reach the intended learning outcomes. And for those of you who are more interested in reading about the Carpentry's guiding principles, I added the link here in the slide. If you think this sounds interesting, we very much invite you to join us. You're very welcome to contribute to the lessons. And I would recommend to take a look at the material that we developed for the workshop that we gave earlier today here at BioC 2021. You can join our Slack channel, Education and Training, in the Bioconductor Slack workspace. In there, there's also a pinned message with a link to our organizational Google Doc, which has meeting notes and information on how to join our Google group. So this group is where we send announcements about our monthly meetings, which currently take place once a month on the second Monday in each month. And of course, if you have any thoughts or ideas, feel free to get in touch with any of us. And with that, I would like to round off and thank you very much for your attention. And I'm happy to take any questions.
Thanks, Charlotte, for a really great introduction to the work of the Bioconductor Education Committee. I think we're holding questions over to the end. So if you're happy to wait, feel free, people put questions in the Q&A tab. I'm wondering, Charlotte, if there's a link to the slides that maybe can go into the, or the incubator resources that can go into the Q&A.
Yes.
People can have a look at while we're going through the other talks.
Yeah, I'll post that in the chat.
Thank you. Okay, our next speaker is going to give us a talk about BioCSwirl, interactive art tutorials for bioinformatics. The speaker is Lisa Cow, and she's joining us from Simon Fraser University. Welcome, Lisa. Looking forward to your talk.
Thank you.
Hi, everybody. Welcome to the BioSwirl Project's Lightning Talk. I'm very excited to be introducing you all today to our R package. So essentially, our R package builds entirely on the Swirl infrastructure and course format. Swirl is an R package where you can generate different R courses. And so R courses particularly cover just bioconductor workflows and analyses and different packages as well. So these courses are done entirely through your R console. It's very similar to, say, a data camp course if you've ever done anything like that. And we started basically as a hackathon project that's been going for about 2 years now. So I wanted to get right into the footage so you can see kind of what the course is like and what it's about. So I increase the quality here. So currently we have students installing our courses through DevTools. Basically, you just install the R package using the install GitHub function. And one of our main dependencies is, of course, Swirl. And so you can see here that we have a variety of courses. The student is currently going through the RNA-seq course, and it lists the lessons. All of the inherent Swirl functionality also works in BioSwirl. So if you've ever done a Swirl course, very similar, you'll be able to jump right in. And you can see that we have different students at the first lesson of each course, installing the different dependencies and learning how to install packages with BIOC Manager. Of course, if you have the dependencies already, we're just going to load it in the background for you. And if you skip it, we'll be running the commands in the background so that you don't have to worry too much about running into errors, for instance. So if we go into what an actual course might look like, this is our PCA lesson in the RNA-seq course. So currently the student is trying to generate a label matrix just to use for the heat map mapping, just so that they have the labels. And you can see here that the student has run into like a very small typo. Basically, they kind of did their first intuition, which was use a function called matrix. However, because the actual syntax is as.matrix, you can see that we have given them a wrong answer and instead suggested the correct answer. And through that adjustment, they're able to do what was needed. If we skip a little bit more forward here, we're just getting them to go through the vignettes so that they know what we're using. Currently, our course incorporates pHeatMap. And now they're going to be trying to generate their first heat map. You can see here that their first instinct again was to use the pHeatMap function. And then as they're going through, you can see that in this particular case, we have given them the actual command to run. And so this is typically because, with a long command such as this one, it can span multiple lines. There's, sometimes typos that occur or bracketing errors. And so students oftentimes find it very nice to be able to directly compare the answer. And this is really nice as well, just because, it helps pad some of that anxiety as well. So the student is going to now try and, I guess, copy and paste the answer to the question, but it didn't necessarily work. So now they've got a heat map kind of going, but it's obviously not very nice. It's not what we're looking for so far. So they're making adjustments as they're going along. Our error messages are going to be popping up, but we're also going to be giving them hints as well. So this is sort of like a guided troubleshooting, if you will. And this will be accommodated to kind of their level. So now they've got the heat map to display correctly. However, they're missing the labels, so they're having to do some adjustments. And now you can see that they have exactly what we were looking for, and they're going to be asked to interpret it. So because the view is a little bit small, they're going to make it larger. And now they're going to be looking at this plot and answering some of the questions that we've given them. All of our courses use standardized data sets that are publicly available, and we're trying to pull information that's already pretty readily available online. We're not trying to reinvent the wheel. Most of the aim is to teach them, why or how we use certain functions and what the workflow is at more of like a holistic level instead of just giving them code to copy and paste. This is really helpful if you're trying to learn an analysis for the first time, particularly to apply to your own data, right? So next, we're going to be continuing on with this lesson. And we're just going to be showing what it's like to learn some of the more statistical concepts and theoretical concepts. Currently, we're having the student here go through DESeq2. As you can see, the environment has been set up for the student. This is usually done as they're going through the course, just as they're running this R code. But say if a student wants to skip ahead to a particular lesson, they don't want to do the first 3 lessons or they've done it before at some other time, then they don't have to worry about having to backtrack constantly. And it also helps us keep a controlled environment so that we can work on that troubleshooting a little bit more. So right now, they're going to be working with DESeq objects and playing around with the results table a little bit and seeing, what it is exactly and how to manipulate these different data frames that they're being given. So we skip ahead here a little bit. We can see that the information is also provided in bite-sized chunks. And this is really helpful when you're trying to wrap your head around some of these new concepts, and it makes it very easy to kind of jump in and feel like the pacing is continuing on. So now that we've got a nice little results table, we're going to be asking the student to interpret some of these numbers that they're seeing, as well as reading this column. All right. So now that they're working with summary objects, you can see that obviously this kind of footage is sped up a little bit because it would be too slow to do it in real time, but it is entirely self-paced. Now they're going to be filtering these results for significant p-values just so that they can actually get to the stuff that we want, right? Which is, which genes are more expressed or which genes are, less expressed throughout the experimental conditions, and they're adjusting for different p-values. I think that one of the really interesting things here to look at, of course, is that, we have this positive feedback that's coming in, and that the student is able to really, not have to read this giant body of text and kind of, have all this theoretical information that's floating around in their head. They instead can kind of do it and break it up and have these hands-on components, which make it a lot more fun for active learning. So now the student is just going to be taking away their results and interpreting them. So I'm going to be moving on to the next few slides now that you know exactly what it is that BioSwirl looks like if you've never been exposed to a particular course like this. So one of the things that's really interesting about BioSwirl is that it's asynchronous, but we're trying to incorporate a lot of these active feedback components. Of course, the gold standard for learning these sort of workflows is workshops, right? But workshops can be, a little bit disruptive to your schedule, and it can also be very expensive. So we wanted to create an open and free option that's very easily updatable, that the community can kind of contribute to in a lot of different ways. And that's something that you might find a little bit different from, say, a book down tutorial, for instance. Of course, our courses are meant for people who are new to coding, particularly those who are coming from, biological or wet lab backgrounds, and who maybe have a little bit of code anxiety too. And we want to make this information accessible just in case, somebody has funding difficulties. Other applications that we've used for this particular format has been hybrid workshops. These have been really nice, part lecture, part hands-on. We also think that undergraduate courses could really benefit from, something like this. As well, we think that down the line, creating interactive documentation for packages would be really helpful as well. So these are our current course offerings, all in sort of different stages of development. I would say our most developed course so far is the RNA-seq workflow, and all of these courses can be done independently of each other. We do want to, of course, add more down the line. And I also want to thank the development team who've been working so hard over the last 2 years. That's me right over here. And we're always looking for new developers. If you're interested in getting involved at all, please do not hesitate to reach out. We are a really nice team, and we're very keen on having beginners who want to get involved in open source as well. So you can find us through these links. On Twitter, we are at BioCTools. Our GitHub repository is under the organization BioSwirl Dev Team. And our website is bioinformaticstutorials.github.io. So thank you so much, and hopefully we can see you soon.
Thanks, Lisa. That was awesome, and sorry for mispronouncing your package name, BioSwirl. I'll get it right.
Thank you.
So yeah, as for the previous speaker, please put any questions you have in the Q&A. And Lisa, if there are any links you want to share with participants, feel free to put them in there as well, so people can find some of those resources in case they didn't have time to type quickly while the talk was going on. Well, it's my great pleasure to introduce the final speaker in this session, Nicole Kramer, who's based at UNC at Chapel Hill. So Nicole is going to be speaking to us about BentoBox, a new paradigm for multi-figure coordinate-based plotting in R. Welcome, Nicole. Looking forward to your talk.
Kramer, and I'm a bioinformatics and computational biology grad student at UNC Chapel Hill. Today, I'm going to be telling you about BentoBox, an R package we've developed that introduces a new paradigm for multi-figure coordinate-based plotting in R. So first, I want to talk a little bit about why we decided to build this package. Even though we have great genomic browsers and programmatic libraries and tools like JuiceBox, HiGlass, IGV, ggplot, gviz, these tools aren't as great for building a complex multi-panel figure like the one I'm showing you here. So here we're not only combining different data types like Hi-C data, signal track data, gene data, we're also looking at two completely different cell types, and this region of data is corresponding to some kind of bioinformatic data analysis that we're doing in R. So in order to put this figure together, we're probably getting its elements from a variety of places. We probably have a bunch of different genomic browsers open, we probably have R open and we're making a plot there, and we probably have Adobe Illustrator open to ultimately customize and arrange all of the elements of this plot. So let's say we wanted to show someone exactly how we made this figure, or maybe we just wanted to go back and change the region we were looking at slightly. This wouldn't really be an easy task because we have all these browsers with all of our information open. So we wouldn't be able to just pass someone some instructions for making that figure. So we thought that there had to be an easier way to make figures like this beyond existing browsers, programmatic libraries, and graphic design software. Something that would let us make and arrange all of our plots in just one place. Something that would extend the rigor and reproducibility of scientific data analysis to its visualizations as well, by being completely programmatic, customizable like existing R programmatic libraries, efficient for visualizing large genomic data sets that we're working with, and user-friendly for both experienced and inexperienced R users. And so we have developed a package called BentoBox that allows for coordinate-based plotting in R, where plots can be programmatically made and arranged on a user-defined page layout with common units of measurement, like inches or centimeters. BentoBox allows us to translate an exact blueprint of our desired plot arrangement into a precise publication quality figure, all within the R environment. So let's go through the process of building a genomic figure with BentoBox. First, BentoBox allows us to create a page in our exact size and the units of our choice. Here we've made a page that is 7 inches wide, 4.25 inches high, and it's all in inches. BentoBox also adds these helpful page guides here to guide you in the building process of your plot. Then when we go to make our genomic plots, not only are we defining the data, but we're also going to be able to define the width, height, and placement coordinates of our plots as we build a complex arrangement of them on our page. So here, for example, we're making a Hi-C heat map that is exactly 2.75 inches wide, 2.75 inches high, and its top left corner is placed at exactly an X coordinate of 0.25 inches and a Y coordinate of 0.5 inches. BentoBox is modular and separates plots and plot annotations for the ultimate control of all the plot elements. So for example, here we can pass in our Hi-C plot object from before, and we can add a heat map legend annotation for our Hi-C plot, but we can also control its dimensions and coordinates on the page as well. As we build up our plot, we can also add many other elements. So for example, here we can add a check of genes in our genomic region of interest. This data is acquired through BioConductor-TXDB packages. And below this collection of data, we can also add a genomic label annotation to give context to what region we're looking at. And beyond looking at single lined up tracks of genomic data like this, which you'd probably typically see in a genomic browser, we can also add other various plot elements to our page in different locations, sizes, and representations. So for example, we can add a chromosome ideogram with cytoband staining information acquired through Annotation Hub. We can now easily visualize and compare genomic data from two entirely different cell lines, with these objects not really interfering with each other. We can continue to build up our collection of data with signal tracks from data like RNA-seq, ChIP-seq, ATAC-seq, and because BentoBox allows us to place these tracks at completely controlled widths, heights, and locations, we can always ensure that we're accurately comparing our data along both axes. So here we can make sure we're comparing it accurately along the y-axis for some kind of score comparison, and along the x-axis here to compare data in the same genomic region. We can even go back to various sections of our plot and add additional elements that you probably wouldn't add in R, like text, for example. So here we're adding our panel labels and our signal track labels and a nice text label for chromosome 21. Now, when we're done building up and tweaking this plot, we can go ahead and remove all the guides from our page with a simple function call. And now our multi-panel genomic figure is ready to be exported and used in publications or presentations, just like this one. And because we made this entire figure with code, we can easily integrate any bioinformatics analysis we did with our data, as well as the visualization, into just one R script. So now if we share our data and our code, our visualizations are entirely reproducible from raw data to final visualization. Another benefit of making visualizations programmatically is that now we can easily change the genomic regions we're visualizing with little additional effort. So instead of going back to a bunch of different browser windows, we can now just put it in a line of code and pass it to all of our functions. In BentoBox, we've conveniently developed this BB_params object that will allow you to store various BentoBox parameters and then pass that into multiple function calls to streamline your code. So for example, if you're looking at one genomic region and you're passing that same region into a bunch of different function calls, you can store that in the BB_params object. This BB_params object is also really handy because it can be used to find the genomic coordinates of a gene if you're not really sure what those coordinates are. So you just pass in the gene name and it'll find the genomic coordinates with some kind of buffer and then pass that region information into all your BentoBox plotting functions. BentoBox is optimized to support various genomic data types and can plot some data types in different representations. None of its genomic parameters are hard-coded, so these can all be customized completely for different genomic assemblies and whatever other features you want about the plots. These functions can also really easily interface with various bioconductor data classes and packages, so it's easy to consolidate our genomic data analysis with the visualization. And because these data are often so large, we've incorporated scale-aware logic into certain functions so that they'll behave slightly differently and more efficiently depending on the size of the genomic region we're looking at. So for example, if we're looking at a 300 base pair region and we have a lot of space to look at, we'll be able to see distinguishing details in gene structures. Like here you see a UTR, you see an exon, you see a gene label. And then when we labeled our region, we'll also be able to see colored boxes representing the nucleotides in this small region. However, when we scale up and look at an entire chromosome with way more information, here we're going to condense our gene structures, remove additional details, and only label the names of the most cited genes on the chromosome so all the gene labels aren't overlapping each other. And now the region is too large to show us any nucleotide information about the region, so we don't show that. Overall, this just makes our code way more efficient and make sure it's handling genomic data types appropriately. And BentoBox's uses don't actually stop with genomics. So although there are existing packages to arrange plots in R, none are as precise and specifically tailored for a multi-plot environment like BentoBox. So in particular, none are actually based on absolute coordinates and dimensions in a specific unit like inches. So here, for example, we've made a complex arrangement of ggplots in BentoBox, and we've gone beyond a simple grid layout where we're overlapping some Venn diagrams, overlapping pie charts onto some line graphs, just something that wouldn't really be simple and straightforward with typical existing packages. This makes it easy to integrate other kinds of plots into your BentoBox layouts, and it makes it useful for genomicists and non-genomicists alike because everyone's going to want to arrange their complex arrangements of plots. If you're interested in trying out BentoBox, I encourage you to visit the BentoBox website to explore the documentation and examples that'll show you how to make beautiful plots like the ones I've shown you today. And I hope BentoBox will change the paradigms of how we make and arrange plots in R, making them just as reproducible as the rest of our data analysis workflows. Thank you so much for your time, and I really hope you try using BentoBox with your data.
Amazing work. Thanks, Nicole. Hopefully we can all ditch, what's it called? Illustrator, which is very challenging to use in place and have BentoBox instead. Great. So I don't know if we can bring all the speakers to the stage, but I'm looking for questions now. Q&A. Hopefully I can find some. All right. Thank you someone for selecting a question. Lisa, can we make our own course packages from Sean Taylor? Great question.
Yeah, this is an awesome question. Of course you can. And I would encourage, if you're interested in kind of forking BioCSwirl and doing something on your own with it, do it. I mean, it's pretty open source. I highly recommend that you try it. One thing that I will add is that throughout the learning curve of adopting Swirl for BioCSwirl, we've noticed that there's been small quirks that have kind of popped up in terms of like, hey, like this is a little bit different, this needs a bit of a work. So if you do want to make your own course, I do suggest, when you hit a bug, you can get in touch with us if you want to. One really exciting thing that we have right now is actually a GitHub Actions pipeline. So if you have a particular course that you don't want to do the manual test yourself, because I guarantee you, first person perspective, it sucks to test courses. We want to be able to have, that automated integration for you to throw your course into. So yeah, we definitely encourage it. So any sort of course that you make in Swirl, you can make with BioSwirl.
Thanks.
And
another question related to Swirl. Does Swirl work smoothly with via piping, non-standard evaluation?
Right to end there. Yeah, so we've noticed that what ends up happening sometimes is when the multi-line part of piping comes into play, it can mess it up. And the reason why this is because all of the lessons are actually stored in YAML format. YAML doesn't really play with special characters much. So if you can keep it all in one line, then it works pretty much as far as I've seen. I don't necessarily know if it works with like the very new pipes that have come out, but as long as you have your migrate or delayer as a dependency, it will work. So we have used the pipe before and it has worked. You just have to kind of keep it all on one line. So if you want to have like a really complicated multi-line piece of R code, it can get a little bit tricky there.
Sorry, I'm on mute. Here we go. The next question's from Nicole. Will BentoBox also help you pick the correct font size for axes, labels, tick marks, etc.?
Yeah, that's a great question. So right now we have kind of helpful defaults that are based on, say, like a standard page size in inches, but obviously that could vary a lot depending on what figure you want to make. So there are some defaults that'll help you, but ultimately we're trying to help you get complete control over the exact size of everything you want. And hopefully in BentoBox version 2, we'll also have some kind of pre-laid out defaults that'll also aid in that process.
Another question for Nicole. Why do you think it took so long for someone to develop such a cool tool? And what were the bottlenecks?
It's a great question. I don't know, but I'm glad because now I got a chance to do it. Honestly, probably a big part of it is the fact that it is so time consuming because by the way that we built BentoBox is based off of grid graphics. And so that's a really low level plotting system, right? So you can't just say like plot and it'll make the actual plots for you. You need to build up every element. So every function that we've written for BentoBox, we've gone in and we're like making those tiny rectangles for the pixels, we're making the dots, we're making everything. So it is really time consuming and a labor of love. So maybe no one wanted to do it just yet.
All right, we have a couple of questions for Charlotte now. So do you keep track of pain points of the courses across sessions? Mike's taught a lot of workshops, but didn't systematically keep track of these things.
Yeah, so that's a great point, and it's something we should definitely do. And it is kind of built into the carpentry's framework. Let's say the repositories have a file for that. So there is a file with instructor notes in this repository. So I think that would be the place to put it, and then it would be available to whoever would teach the course in the future. So that's definitely a great point.
I had another question, Charlotte, which is you mentioned that more people, more community members might potentially be able to get some carpentry training to become accredited trainers. Do you have a sense of that, how that scales, or like what the typical number of trainers per year might be, and what the process would be for people potentially applying? Is it like an expression of interest, or yeah?
Yeah, I think that's what we have in mind. So that's what we did for last year. So we kind of made a call out for people who might be interested, and then well, whoever was available at the time, then there was a selection there, and of course how many seats were available in that particular online course. So I think that's how we will do it. We'll make a call to the community for whoever might be interested. For the number, it kind of depends a little bit on, we have some funding applications in as well. So, but yeah, maybe something like 5 or so again for next year would be a reasonable number.
And I kind of wonder, seeing Lisa's talk with BioCSwirl and hearing your talk, Charlotte, are the two approaches quite distinct? I guess, is one more like for people doing a course independently, whereas the others, or do you think there can be some, BioCSwirl could be used to kind of deliver some carpentry's material? Or I don't know. That's a naive question, but
it's a great question. We already invited Lisa to present in our meeting, and she did that. So we have already set up the connection, let's say. And I think it's definitely possible. I mean, that the BioCSwirl can be used to deliver or for people to get access to material. So we also found it very interesting approach.
I loved the little encouraging messages, oh, you're nearly there, or all that work is paying off. That's very cool. We have another question of Nicole, which is, here we go. What's on the horizon for BentoBox 2? People are already so excited about BentoBox 1. They want more, Nicole.
Yeah, just the work never stops. We have a lot of great ideas for BentoBox 2. Some of it, I don't know how interesting other people would find, but we want to do a lot of infrastructure changes and like stronger S4 class integration, more bioconductor integration, make it even stronger there. But we also want to do some fun stuff for usability. So some cool multi-plotting functions, maybe some fun templates for people, more helpful defaults. Grammar of graphics style implementation is a really big thing we want to start doing. So yeah, we have a lot of great ideas.
I imagine it'd be an awesome tool for data journalist type people as well. And is there ways to kind of reach them to make it, okay, it's great for bioconductor applications, but plenty of non-bioconductor applications?
Yeah, I mean, I think honestly, one of the greatest parts about BentoBox is just how easy it is to even combine normal plots you'd make in ggplot or baseplot or other things like that. So I think if we come up with more beautiful figures like the one that I showed at the end of the talk, we'll get more data scientists on board to just use BentoBox to make really cool figure arrangements of normal data representations.
Great, thanks. Are there any further questions? Feel free to raise your hand if you want to ask in person. I think the ones here are answered in the chat. So if not, it just remains to thank all of our fabulous speakers in this session, Charlotte, Lisa, and Nicole. And Yeah. See you at the next session.